once lipids are broken down by esterases, the products formed pose a major challenge. These molecules have two ends, one water-loving or hydrophilic and the other fat-loving or lipophilic. In other words, these molecules are amphipathic. The hydrophilic ends dissolve in the watery environment of the gut. But what about the large lipophilic tails that are hydrophobic? The solution is a structure where the hydrophobic tails form the core such that only the hydrophilic ends face the watery environment outside. This might solve the problem of solubility. But unfortunately, these structures are unstable in the intestinal environment and may coalesce into larger aggregates that cannot be efficiently absorbed. The body has a brilliant solution to this problem, its own amphipathic molecules, the bile salts. When bile salts reach a certain concentration, they spontaneously self-assemble, such that the hydrophilic ends face outwards and their lipophilic ends inwards, creating a protected core. This structure, called a micelle, is pretty stable and forms a framework to integrate the products of lipid breakdown. The fatty acids, monoglycerides, and cholesterol are integrated such that the hydrophobic ends are hidden in the core while the hydrophilic ends face outwards. This hybrid structure is called a mixed micelle. The mixed micelle can transport the lipid breakdown products through the aqueous environment of the intestine to the surface of the intestinal cells. But what happens once the micelle reaches the intestinal cell surface? That's next. Subscribe and hit the bell so that you don't miss it.